My name is Rebecca Laborde, and I'm a Postdoctoral Research Fellow at Mayo Clinic in Rochester, Minnesota. I am discussing an article written by the Departments of Otorhinolaryngology and Experimental Medicine and Pathology entitled Transcriptional Profiling by Sequencing of Oropharyngeal Cancer, which will be appearing in an upcoming edition of the Mayo Clinic Proceedings. This study involved the use of massively parallel DNA sequencing, which is also called next-generation sequencing, that was used to interrogate the transcriptional profiles of a subset of head and neck cancers using mRNA-seq. Head and neck cancers traditionally occur in patients greater than 60 years of age that have a history of smoking and/or drinking. Exposure to alcohol and tobacco are generally considered to be the most significant risk factors for developing squamous cell carcinomas of the head and neck. However, the incidence of oropharyngeal squamous cell carcinoma has been dramatically increasing in younger patients that lack exposure to tobacco and alcohol. Mounting evidence points towards an association of human papillomavirus infection in this patient group, primarily HPV types 16 and 18. As patient treatment strategies are developed that focus on a more individualized treatment approach based on the molecular aspects of tumor biology, it becomes critical that we correctly stratify patients that present with multiple risk factors. It's important that we develop molecular tools to accurately predict which risk factors are most significantly influencing the development of a patient's disease, such as tobacco and alcohol exposure or the presence of a high-risk HPV infection. To approach this question, we generated and analyzed full transcriptome sequencing for a total of 9 oropharyngeal tumor and matched normal patient samples to characterize gene expression patterns. Initial findings were validated using a larger group of 38 patient sample pairs and a digital counting technology. The patients included in the study represented 3 groupings of risk factor exposure: current smokers with a greater than 2-pack per week smoking history, a current smoking history, former smokers with a smoking cessation history greater than 10 years, and never smokers who lack a smoking history. The HPV status of these groups varied but was determined by multiple testing procedures. Initially, we compared the global gene expression patterns of these samples when grouped by smoking history. We observed, as expected, that the gene expression patterns of current smoker tumor samples were different from those of never smoker tumors. An interesting observation was that the expression profile of tumor samples from former smokers more closely represented that of never smokers than current smokers. Also, that the normal tissue of all 3 groups showed similar patterns of gene expression. We then identified specific gene targets that displayed differential patterns of expression in samples divided by smoking status. We grouped the differentially expressed genes into pathways and focused on genes within the p53 signaling pathway. Following the validation experiments using a larger group of patient samples, we identified a significant expression pattern between smoking groups for 2 specific DNA damage responsive genes in the p53 pathway, ATR and CHEK2. We found that expression of ATR is significantly upregulated in current smoking patients as compared to either never smokers or former smokers. The expression of ATR is indistinguishable between never and former smokers. Further analysis that included the HPV status of these patients found that both genes ATR and CHEK2 are significantly upregulated in current smoking patients that lack an HPV infection. These findings hold immediate significance for patients because they provide further evidence of the benefits of smoking cessation and that they occur even at the molecular level. This should provide additional incentive for adhering to a smoking cessation program. These findings are significant for clinical practice as the pattern suggests that significant patterns of smoking cessation may allow for RNA transcript regulation to return to patterns more closely resembling that of non-smokers. This would suggest that stratification of patients based on smoking history should consider former smokers as a separate group, rather than including them with either current or never smoking patients. Our ongoing research is focusing on identifying the mechanisms that drive transcript dysregulation that was observed in this study. This will include a careful inspection of the genome to identify changes to the DNA such as mutations, and analysis of the epigenome to identify alterations such as DNA methylation. It is likely that both of these mechanisms contribute to the genomic changes that drive the development of oropharyngeal cancers. Finally, this work demonstrates the potential of transcriptome sequencing to become an important clinical tool as we move toward a more individualized approach to patient care. Improvements in sequence output and a decrease in costs allow us to consider this as an option for developing tools to more accurately stratify patients and define molecular characteristics of an individual's tumor biology. These advancements have the potential to supplement current treatment approaches and ultimately to improve patient care.
We hope you benefited from this presentation based on the content of Mayo Clinic Proceedings. Our journal's mission is to promote the best interests of patients by advancing the knowledge and professionalism of the physician community. If you're interested in more information about Mayo Clinic Proceedings, visit our website at www.mayoclinicproceedings.org. There you will find additional videos on our YouTube channel, and you can follow us on Twitter. For more information on healthcare at Mayo Clinic, please visit www.mayoclinic.org. This video content is copyrighted by Mayo Foundation for Medical Education and Research.